How to use the model organism databases. At the top of gene function table, there's a box that you can check whether you want to see only the ortholog candidates with the highest DIOPT score or not. You may want to uncheck this box if there is likely more than one ortholog in your model organism of interest. However, one needs to be careful when using this feature since genes with a low DIOPT score may not be true orthologs of the human gene of interest. We provide the following information for 7 organisms. First, gene name. This is linked to the gene page on respective model organism databases. If you want to find out more about the phenotypic information and resources available for each model organism, these are the places to go. For example, on FlyBase, there will be a list of all alleles that have been generated and are available from public stock centers. Next, PubMed link. This is a list of publications that relates to the gene of interest. The advantage to using these links, as opposed to searching for the human gene directly in PubMed, is that you may miss literature that uses an old gene alias for the human gene and many model organism literatures where the gene name is not exactly the same. Next, DIOPT score. This is a score of how many ortholog prediction algorithms predict the gene as an ortholog of the human gene of interest. Note that we typically consider a DIOPT score of above 3 to be solid, but there are cases where genuine orthologs only have a DIOPT score of 1. Next, expression. This column lists the tissue where the gene of interest has been reported to be expressed in human or model organism databases. Human gene and protein expression data are from GTEx and Human Protein Atlas, respectively. Some have a button with pop-up link, such as for human and for fly, whereas others are hyperlinked to respective model organism databases. Gene ontology, or GO terms, are filtered by experimental evidence codes and obtained from respective human or model organism databases. Some terms are filtered out, including those with computational analysis evidence codes and electronic annotation evidence codes. Other links, like Monarch, will bring you to the PhenoGrid, a chart that provides a quick comparison between the phenotypes associated with the gene of interest to known human diseases and model organism mutants that have phenotypic overlaps. If your mouse gene has a knockout mouse made or planned by IMPC, the IMPC link will bring you to the page that details the phenotype of the knockout mouse. Another resource that integrates model organism data is Gene2Function. The hyperlink to that tool is located at the bottom of the model organism search page, next to the Power By link. I want to show you some useful links that you can navigate to from the model organism page. The first link is Monarch, and when you click on this link, a page like this will come up. This is a grid-like structure where phenotypes are linked to genes and different model organisms. The next useful link is called IMPC. Here, you will be sent to a page that looks like this. This is a list of phenotypes of the knockout mouse of your gene of interest. Each phenotype has a lot of details that you can click into and explore. And finally, if you click into each of the different model organism links, you will be able to navigate to the respective model organism pages. For example, if you click on this link, you'll be sent to the FlyBase page. This is what the FlyBase page for this gene looks like. You may want to look for different alleles and phenotypes that are available for this gene. You can click here and find that there are multiple alleles available, and these are the phenotypes associated with these alleles. The human gene protein domains box provides predicted protein domains of the human gene. There are some overlaps because we use multiple domain prediction programs here. If you would like to know if a certain human protein domain is conserved across species, you can scroll down to the multiple protein alignment box. And here you can enter the amino acids of interest, and they will be highlighted in blue. We are currently developing features to convert the genomic variant information into protein variant information, but until this feature is installed, please manually find the amino acid of interest using these fields. If the amino acid that you highlight based on the number is not the one you were expecting, you may be using a different splicing isoform. You can use the UCSC browser feature mentioned before to find out whether your protein of interest have multiple splicing forms and identify that the one that is used in the multiple protein alignment. In principle, we use the longest isoform to display in this box. You can do the same for other amino acids of interest and for other organisms by clicking the check boxes next to the model organism initials.